Hello everyone, welcome to another series of VPH Institute Keynote Webinar. My name is Mojtaba. I'm a postdoctoral researcher at the University of Oxford and a member of the Young Scientific Committee at VPH Institute who organized this series of the webinars. I will moderate this webinar and it's my pleasure to have you all here. For today's webinar, I'm honored to have Dr. Ehsan Abadi as a speaker, who will be sharing an exciting story about visual imaging trial and how they use it in the quantitative CT. Ehsan is the assistant professor at Duke University with a joint affiliation in the Department of Radiology and Department of Electrical and Computer Engineering. He's also co-lead in the Center for Visual Imaging Trial. For today's webinar, as usual, we have two sessions. First, speaker's presentation for 45 to 50 minutes, and following you with a Q&A session for 10 to 15 minutes. If you have any question, please do type them in the either chat box or question box you can find in right-hand side of your screen. I think now we are ready to start. Ehsan, thank you again for joining us, and please, the screen is yours.
Sure. Thank you, Mojtaba, and thank you for inviting me. Good morning, and good evening, and good afternoon, everyone. I'm very glad to be here. Today, I'm going to talk about some of our trials, introduce what it means, and with a few introductions on various topics and components of it. And then I'm going to give you a few examples of how that can be used for quantitative CT. Again, there would be lots of application at this platform, but for the sake of time, I thought it would be nice to discuss a few of those applications in quantitative CT. So without further ado, let me get started. Here is my funding disclosure. And before I begin, I would just want to acknowledge that the work that I'm presenting is a result of collaborations with great colleagues at Duke University. Many of these work have been work of them, and I'm presenting on their behalf. So in advance, thanks to my colleagues at Duke University. So let's just start with imaging technology. It is advancing, and it's advancing at a very high pace. If I want to give you an example on CT scanner, there has been a lot of new technology and new features of these CT scanners. I'm just giving you some examples, and you see as we're getting to the closer to 2023, there are more technologies that are coming into clinical practice. Namely, the main one that is very hot topic right now is the photon counting CT scanner that every major vendor are pursuing prototypes and scanners for it. So this high pace of technology advancement is outpacing our ability to evaluate these technologies and hardware and software, and to optimize them, to make sure that we are using it in the right way for the design and clinical use. So if you want to do that, our objective is to evaluate and optimize current and imaging technologies for quantitative characterization of the diseases. That is what we want to do. But how do we do this? How do we evaluate and optimize? Well, the first step would be, let's use physical phantoms. We have these phantoms. We image them across multiple settings or with a new technology, and we see what's going on. And we use that as a metric to evaluate and find optimum parameters. There are limitations with it. These physical phantoms are usually not anthropomorphic. They do not have the shape of human beings, doesn't have that anatomical complexity. And even if you do have one, let's say you have a physical phantom that is anthropomorphic, they're not disease specific. They're not reflecting particular diseases that might be of interest. And they're not diverse. It would be one. But you want to have a solution that works for a population of patients, not just for one patient. So how to address this would be, let's use clinical trials. If I have patient images, then that would be great. I don't need to be worried about the diversity or anthropomorphism or disease. But the issue with clinical trials is that often they are ground truth limited. So you image a patient, but you don't exactly know what's going on underlying. If the patient has a disease, you have a limited access to that ground truth of the patient, unless you do biopsy. And even if you do the biopsy, you may not be able to see the full picture. So there's a ground truth limitation. These trials are often very expensive. It takes a lot of money and effort to run these trials. There's a dose concern. If you want to image and optimize a patient across various scanners or multiple settings, you need to overdose the patient, and this is something that we don't want to do. So that's also ethically infeasible. And the scanner availability is another issue for these new technologies, and not every hospital has access to that. So we cannot also do those studies as well. But that all brings us to the reason and rationale of why we do virtual trials. Because with virtual trials, which is, can we mimic the clinical trials in a virtual space? You would have access to ground truth. You know what you're modeling. It would be cheaper. You're working with simulations and computers, not multi-million-dollar scanners. These are virtual patients, so you do not need to be worried about those concerns or imaging them multiple times. And as far as you have the models for them, you can experiment anything that you want, and you can design any studies that you would like to. So that's what brings us to the domain of virtual trials. How does it work? You basically mimic a real clinical trial. You have population of patients, virtual ones, and then you target them based on the cohort that you want to study, anatomically and physiologically. And then you also need to model the scanner virtually. So the scanner or the technology that you want to look into, you would model that both in terms of physics and geometry and acquisition and post-processing, all that. And then you do assessments. You do the experiments, the question that you want to answer. You use these virtual images to address those questions. So that's the basic framework of doing virtual imaging trials. So I'm going to go over the main two components, which are the virtual patients and virtual scanners. I'm going to introduce them to you, a few examples of what we have done in the recent few years, how these models look like, how they're developed. And in the end, I'm going to give you a few examples of how we have used them recently, as I mentioned, for quantitative CT. Before going there, let me just also introduce you that we have a Center for Visual Imaging Trial at Duke University. This is an NIH-funded center. We do have lots of development and resources for doing virtual trials. If you go to our website, you can see we have a monthly forum webinar that talks about these topics. We have resources, software to share, data to share. So please check out our website, and if you're interested to know more about this topic. So let me go to the virtual patient side. This is not a new topic. Over 50, 60 years, there have been work on creating mathematical models that look anthropomorphic. And over the years, they have been improved and enhanced to be more and more anthropomorphic. We are one of the groups that work with virtual phantoms, and we create these whole body anatomy phantoms called XCAD phantom. These are based on CT images or MRI images, and we get the major organs and major structures, and we create a library of them, including muscular system, vasculature system, skeleton, and so forth. The way it works is that we get patient CT images, as I said, or MR images. We automatically segment the major organs. For the organs that we are not able to segment, we use our Atlas library of existing phantoms, and we use registration algorithms to complete the organs that we did not segment, and that would give us a new existing computational phantom. So that is the basic way of modeling this. But this would be an approach that would create and model major organs and anatomy of the patients. And you also want to have a diverse army of these. So we create models for adults, for pregnant, for pediatrics, and that will help us to target specific cohorts in a particular study that we want to perform. One limitation with this approach is that, so with this, you only get the major organs, but what about intra-organ structures? You don't get them necessarily from a CT or MRI image because there is a limited resolution and limited capacity of what you can segment. So our approach has been that for the structures that we don't see, we use mathematical models to create those structures, but we do so in a way that is anatomically informed. Either anatomically informed based on morphometry studies. There are work that they measure shape, size of different structures, and we look at that and try to mimic that path. And there is another way that you would get access to a higher resolution data, let's say micro CT images or pathology images, and you use that as a resource to create something that looks realistic, but also create these intra-organ structures. So we have done this for multiple organs. Here I'm showing you a result for modeling intra-organs of lungs. You see airways and arteries and veins that we have mathematically modeled. You can create lung parenchyma, the secondary lobules or other components inside the lung that are not vasculature or airway system. We have done this for trabecular bone, bone marrow. Also, we have done this work for kidney vasculature or liver vasculature as well. So this is another area that needs to be considered when you're creating your virtual phantoms. The other aspect is motion. So often patients move during the scan, so we need to model that. Particularly the cardiac motion is very important because when you image the patient, cardiac motion is always there, and often it creates artifacts that you want to assess and you want to minimize. So if you want to do a realistic scan, you also want to have a phantom that has motion in the heart. Also, there are patients who cannot comply with holding their breath, which is a typical protocol in CT scans. So with that, you also need to model a respiration as well. So we have done this using four-dimensional volumetric cases. We have created these phantoms that have motions in them. Here I'm showing you a result of one of our computational phantoms. So this is a phantom at a CT image. It just represents in an idealized CT image, and you can see how the respiration and cardiac motion is looking. This is parameterized, so we can change the heart rate, or we can change the respiration rate or amount of respiration, which that helps us to study various magnitudes of motion and look at their artifacts in CT images. And often we do these imaging experiments for a particular disease or for a particular application. So another important aspect is to model the diseases that you want to study. So we have worked on various disease modeling. Here I'm showing you a few examples on cardiac plaques, COVID pneumonia, COPD or emphysema in particular, and bronchitis, and liver lesions. Basically, these models are added to these phantoms that we create. The models come from three-dimensional data, again, either from real CT images, if we can segment them, or from pathology or from micro CT images, if we do not have access to the ground truth of them through volumetric images. And then we do this for a variety of them to make sure they're representative. So we are not creating only one lesion or one demonstration of emphysema, but rather we create this variable rendition that would give you different extent, different size, different distribution, different material, so that you are basically mimicking the representation and the diversity that you would see in your clinical practice. So disease modeling is also important, and with this, you would have a population of patients virtually, which you can use to study. But what is the next step? that we need. We need also virtual scanner because this is an imaging trial study. So we also need to create visual scanners. And how does that work? So for visual scanners, the main idea is that you would create the acquisition system first. So for the scanner that you're modeling, what is the geometry of the acquisition? What is the physics of the source or the detector? Here in particular, we're doing CT, so we need to make sure that we understand the geometry of how X-ray tube is rotating with the detector, how many projections we are taking in one rotation, what is the speed, what are the dimensions, what is the physical size of the detector, if there is anti-scatter grid, how do I model that, if there is any motion in the X-ray source, how do I model that, what are the materials of the detector, what is the spectrum on the X-ray source, and all that. So this is the first step that you need to understand, the acquisition system of the scanner that you're modeling. And you often want to be as scanner specific as possible because you want to answer questions that matters to the clinic. The scanners that are in the clinic are the ones that you're interested to look into and optimize and evaluate. So once you get that, once you have a good model of your acquisition system, then you also need to have a model of simulations. And that means that for the sake of X-ray scanners, let's say, then within that acquisition geometry, you would have photons that would come to the patient, interact with it, and then go and hit the detector and get some signals. So with that, you need to be able to simulate what happens with these photons. There are, again, in the case of CT, that's also applicable to other modalities, but has a different physics and interactions. But in the case of CT, you have X-rays. Some of them would be attenuated in the patients, depending on their body and then the path. Some of them would get through and hit the detector. Some of these photons get scattered. You also want to simulate dose, how much dose would be absorbed by the patient. So these are, you can do this through Monte Carlo approaches, through other ray tracing algorithms that would help you to calculate all these interactions. And then once you have that, once you have the signal, then you need to form this image. Again, for the sake of CT, there would be CT sinograms, projection images that you need to calculate, and you would try to make it as close as what you see in a clinical scanner. So the way that the images are being pre-processed in a clinical scanner, you would mimic that approach, beam hardening correction, let's say there are scatter correction, there are other calibrations for water and so forth, and then you try to mimic that. And then in the end, you reconstruct the image to get the volumetric images from sinograms. And similar to the previous steps, again, you try to make it as close as what you see in the clinical practice. So that would give you a framework to create your visual scanner, and that you can use to image your virtual patients to get CT images. We have modeled such a simulator. We call it DukeSim. This is a GPU-based CT simulator that generates projection images. We have two major engines in it. We have a Monte Carlo that calculates the scatter and dose for us. And we have a ray tracing, which calculates the primary signals at the detector. So with those two, we can calculate the sinogram images. We are modeling various features that are very scanner specific, and these new advanced state-of-the-art scanners are using it. Just to name a few, tube current modulation, which modulates the X-ray flux as it rotates through the body. We are modeling that. We have all these post-processing steps, like beam hardening correction or water correction that we apply. We try to be as realistic as possible, again, in the sense of the geometry and then the attributes of the scanner with all the offsets, and having energy dependent response, modeling both energy integrating detector and photon counting detector and so forth. So this is another, as I said, a step that is very important if you want to answer something clinically, Because if you don't do these things realistically, if they are not mimicking the attributes of the scanner, then you might not be able to predict the outcome in a realistic way. So that's the major reason that we try to be as nuanced as possible in what we are modeling. And then as I said, we want to make sure things are realistic. So we do validations. And the way it works is that we create virtual models of some phantoms, and we image them with our scanner. We have physical versions of these phantom as well. We go to the clinic, we make the same scans, same protocols, same settings, and we compare the images. And that would give us a confidence of how close our simulations are to what you see in the real data acquisition. Here I'm showing you one example that for a particular phantom, the top row is the real data acquisition, the bottom row is our Duxim acquisition. Different columns show you acquisition at different dose levels. We have done this for multiple scanners and for multiple settings. And we have compared them in terms of quantified image quality metrics. Here I'm showing you one example of a particular photon counting scanner that we did last year. From left to right, you see noise magnitude measurements, with red being the real experiment and blue being our simulations. The middle column is a noise power spectrum, reflects the texture of the noise that you see in the image. On the right side is the HU or CT accuracy number, which shows you how close our measurements are in terms of the HU value or the intensity value in the CT image. And the results demonstrate that in terms of image quality, you are very close to what you see in the real experiment. And that gives us confidence that at least for these metrics, we are very realistic and we can use these models of virtual scanners. Of course, the phantom that I showed you in the beginning was a cylindrical simplified phantom. We also want to make sure that this works with more anthropomorphic and more complicated phantoms. The challenge is that having access to that an anthropomorphic physical phantom. So for the cases that we have, we would do these validations. This is one example that I'm showing you a physical phantom that we also model a virtual version of it, and we have done validations for it across various conditions that you see. Again, red is real data, blue is simulated data, and these are for two different energy integrating scanners. The results are HU values you saw, and now I'm showing also the noise magnitude of them. Again, it highlights that even in an anthropomorphic condition, we can replicate a real experiment. So far, I have been discussing the developments and components of a virtual imaging trial. Namely, we discussed virtual phantoms, how they're modeled, how they look like, the diseases that we can model, and the conditions of motions and abnormalities and the population and diversity and so forth. Then the next step was a visual scanner. I showed you how you can model visual scanners, how you would change the physics, how you can be scanner specific, and then how we validate them against experimental measurements to make sure that we are modeling various scanner types and models. So now that I have these tools, I want to demonstrate a few applications in quantitative CT and show you how we have used these tools to address some clinically relevant medical imaging questions. I'm going to focus today on lung imaging, but let me tell you that this application, this virtual imaging trial platform has various applications, from dosimetry to technology optimization. You can optimize the scanners for various settings. You can compare different technologies. You can assess dosimetry to the intubations. You can look for image quality. So there are various applications and also for various diseases. But my example would be only for lung imaging for today's talk. So long quantifications are predictive of disease severity, and that's something that we already know. For years, we have seen that if you measure things in the lung, they are predictive of outcomes. But there's a caveat. We have observed over years that these quantifications are highly sensitive to imaging parameters and scanner mix-up models. So things are predictive, but once you combine images of scanner A and scanner B from institution C and D, then the results do not match very well, because the numbers that you get are reflective of the patient attribute, but also they have some features of the imaging parameters or scanner models that you use. And that's a huge challenge, because if you want to do cross-sectional studies or longitudinal studies, you would have images across various scanner mix-up models. In a longitudinal study, after 5 years, there is a little chance that the patient would be imaged with the exact same scanner settings and parameters. So that's something that we need to address if you want to use quantitative CT for lung imaging in the clinics, basically, in a way that is effective clinically. Let me give you 2 examples of what I mean. Here, I'm showing you an acquisition of the same patient. So there's one patient imaged, and the patient was imaged at 4 different reconstruction settings. Top row is a different kernel, bottom row is another reconstruction kernel. Left column and right column are different in terms of their pixel size and slice thickness. Other than that, it's the exact same patient, exact same acquisition. And density in the lung is a very predictive attribute that we want to quantify. On the right, I'm showing you the plots of the histogram of these images. The histograms are from the same patient, and ideally, they should reflect the disease severity of the patient, not the scanner or the condition. But as you see, the histograms are very different, and the only difference is that you use different imaging conditions. So that's problematic, because as I said, your quantifications are affected and impacted by those settings, and this is something you want to avoid. Let me give you another example. A patient is imaged and emphysema is quantified. This is a way of quantifying emphysema, that you look at a particular threshold. Anything below that, you assign it emphysema. This is a very clinically relevant metric. They have shown this, that has correlation with patient outcome. So that's an important score. When the patient was imaged, a regular dose was used, a smooth kernel was also used. After 5 years, the patient was imaged again. And this time it was imaged with a low dose with a sharp kernel. And as you see, the emphysema score is 26%. 5 years ago, it was 11%. So that's a 15% increase. So the question is that, did the patient progress or not? Is this a progression or not? For this very case, we happen to have also another CT scan of this patient. It was imaged at a regular dose on the same day and reconstructed with a smooth kernel. And that shows 10% of emphysema score, which if you compare with 11%, there is no change. So if I go back and forth, So if I go back and forth, after 5 years, I have 2 acquisitions, gives me 16 different % of results. So how do I decide that? How do I deal with this situation? And how can I use this quantitative data in order to reliably say that did this patient progress or not? So what are the solutions? We have done a few works that I want to demonstrate that would address some of these challenges. And the basic first one is that let's optimize acquisitions. So if there are particular acquisitions that we know that they are not giving you accurate results for any reason, they are skewing or giving artifacts or affecting your measurements in an incorrect way, let's just figure that out and try to avoid them. So you do this scanner specifically because different scanners would have different attributes. But also that needs to be patient informed, because patient attribute may also affect that, and that needs to be considered, that is an optimized protocol necessarily an optimized protocol for everybody, or does it matter what type of patients you're imaging? So that's one approach that we need to consider. Second is that we also need to optimize our quantifications. If the way that we're doing these quantifications are affected by the quality of the image, let's say the noise or the spatial resolution or the artifact, let's try to minimize that effect. And also let's harmonize them. If you have images that are coming from different vendors or different institutions, can we transform them in a domain that they would be harmonized so that the quantifications are, the effects of imaging parameters are minimized, and what you're actually looking at is more reflective of the disease. So basically, can we make them comparable in a sense? So I'm going to cover these few examples of solutions. And again, bear in mind, I'm talking about all of this, but this is a visual imaging trial. So for every single solution that I am providing, I want to also demonstrate to you that we did this with virtual trials, and that was the only way that we could have addressed these questions. So let me start with the protocol optimization piece of it. We created, this is, as I said, the example was in emphysema quantification. So we create this variety of human models with emphysema. As you see here, the yellow regions are reflecting emphysema, and you see there are different shapes and types and extents and distribution of them. And then we image this with our Duxim simulator across various scanners, across various acquisition settings and image processing algorithms. And we compared errors in the lines for quantification and so how those quantifications are affected. And with this, basically, we did some protocol optimization. Here, the y-axis I'm showing is the error. So the larger this value is, that means that that's not a good protocol. The error in the long measurements are actually high. And we have looked at radiation dose, reconstruction algorithm, reconstruction kernel, slice thickness, pixel size. So for the very same patient, we imaged it at various radiation doses, various reconstruction algorithms, and all of that. And with this, we were able to find the optimal protocols. What would be the better reconstruction algorithm to use for this particular study? What would be the better slice thickness to use? What would be the better pixel size to use? And we have identified that. And this is, you can answer this question uniquely with virtual imaging trial because you have the luxury of having a cohort of patients and imaging them 100 or 200 different times. You cannot do that with real patients. So that's the first approach. You optimize your protocols. The other part is that you need to understand how your patient attribute is affecting the quantification. For this example in particular, we are measuring density of the lung. And then usually when you are doing a CT scan, you are advised to inhale. So you are at your full end inspiration. So your lungs at the largest volume, and they would image you. But not everybody would adhere to that, would not comply to that. But if you have a different lung volume, that by itself would change the density of the volume. And then density of volume is what we are measuring. So the amount of inspiration that the patient has would directly affect this. So one thing that we need to understand is that how much of that how much of that variability would affect the quantifications in the final result. So what we have done is that we have had these virtual patients. We have modeled them at different respiration levels. Here I'm showing you one example from 100% respiration to 70% respiration. And then we have done our quantifications. Y-axis is the quantification. X-axis is the lung volume percentage from 70% to 100%. The error bars reflects of using it for different patients. And then so ideally you would have error bars on the y-axis because you have different patients. But in the x-axis, ideally you should not have any changes because, as I said, your quantification should reflect the patient disease, not the volume of the patient, because we are not measuring the volume of patients, but it is affecting it. But then we have the ground truth, and we can see how this is compared against the ground truth. So if you look at the red dots, those are the ground truth values. And if you look at the blue plots, those are reflecting what we have measured. And if you see, as you see that at the 70%, actually this the error measurement is actually pretty bad. So if the lung volume is low, it's not doing a good job of reflecting what you see in the ground truth. And this is a well-known phenomenon. So there are ways of correcting for this. So there's these two other colors are methods that you adjust for this. There are statistical methods, there are physiological methods. But then we can evaluate them because we have the access to the ground truth. So we can say in that example, in this example, we can see that like the physiological model was doing a better job to adjust for this. So if you want to adjust for this effect, we have observed that this particular physiological adjusted model was better in terms of mitigating the effect of lung volume. So these are two examples of how you can do protocol optimization, and you can address the patient attribute variability. The other is that how can we optimize quantification? As I said, if you are doing quantifications, is there a way to make this a little bit more quantified or measuring things more accurately? So we have used our virtual tools in order to segment emphysema. Instead of using a threshold-based modeling that I showed you earlier, can I more reliably measure this? And we have developed a deep learning algorithm that can segment emphysema, but that deep learning algorithm was trained based on our virtual data. Because we have the exact knowledge of the ground truth, we have perfect labels, and that would significantly help us to model these networks with a high performance accuracy. So here I'm showing you the top row is a conventional way of measuring emphysema. The bottom row is our deep learning approach. Different columns are showing you that for the same patient, different conditions. So again, ideally, the numbers should be exactly the same. The blue values are the numbers that you are quantifying. And as you see on the top row, you go from like 4.8% to almost 20% for that patient, which is artificial. The difference is because of the scanner and not the disease. But you see that we have done a pretty good job of making this more stable, that we go from 3 to 3.6 to 3.8 and 6% for this very high energy and low dose scan. So we have shown that this our deep learning approach is helping with our quantification to make it more robust, because it's less susceptible to imaging conditions, and also is reflecting more of the emphysema rather than just randomly selecting some threshold points for the low attenuating regions. And then this is not just with the quantification, we have also shown that it has a clinical meaning into it as well. So when we use our robust quantifications compared to conventional ones, we were able to predict patient outcome better. Here, y-axis is a lung function number. This is the diffusive capacity of lung for carbon monoxide. This is a clinically relevant thing that you need. If you can quantify, that's very important. And blue dots are our approach of doing this with deep learning, and we have seen that we have a major improvement in terms of correlating and predicting this compared to the previous method. So that gives us the confidence that we can actually use this approach to more accurately predict outcomes. So it has this clinical utility in the end. We are not just improving quantifications, we're improving the quantification, and we show that it actually matters in terms of clinical utility. I'm gonna end my talk with my last example. I show you this case. As I said, we have two images and we don't know which one to use after 5 years, which brings us to the idea of that now we need to do harmonizations. So we have developed a physics-based harmonization framework that will transform these images to a reference image quality index so that I can make my measurements, use them in a way to compare thousands of images from different scanners, longitudinally or cross-sectionally. I'm not going to go over the method, but let me just briefly tell you that this is a modular method, and the idea is that we do voxel resampling, we match the spatial resolution attributes and noise attributes of them every image into a unified image quality matrix, and that's what we call our physics-based harmonization. Let me show you a few examples. This is one image. We have low dose and sharp kernel for this patient. At the same time, we have also the regular dose and smooth kernel for this patient. The right image is something that we often want. We want a regular dose, we want a good dose and a low dose, and we want a smooth kernel. This is the desired condition. But sometimes we do not have that access to the right image, and for whatever reason, we have only access to the left image. So if I can harmonize the left one to the right condition that I know is my desired, that would be ideal. So I want to go from the red histogram to the green histogram. So this is the result of after doing my spatial resolution matching, and then that after also doing the noise matching. So you see that now the middle image is my harmonized image using the left one, and is very close to the target regular dose on the right one, and that's promising. So with this, I would be able to compare these images when I have various imaging conditions. We have done this for various cases, and here I'm showing you some quantitative results. This is bland Altman plot that we use as a way to quantitatively demonstrate the consistency of our results. So ideally, because I want to reduce the variability, the y-axis should be 0, because these are different conditions for the same patient. And the x-axis shows the diversity of the data that I have. And here I'm showing you that without doing any harmonization, the left results show how variable my images are. I can go up to like 30% variability, and we're able to bring it down to less than 5%, which is very promising if you want to do multi-institutional studies. And yeah, here I'm showing you some of the results. If you see, like in terms of bias, we went from 7% to 1%, minus 1%, and the limits of agreement are dramatically changed and improved. So what that means? That means that for that very case that I showed you after 5 years, which we had the low dose, if we were up to harmonize it, we would have seen a 10% magnitude of emphysema, which was very close to the regular dose reference data that we had, and that was something that we were able to use for our study and for our longitudinal study in this case. So some taking home points. I demonstrated that virtual imaging trial is a solution to our limitations to conduct in vivo experiments because of the cost, time, ground truth. And it's also a solution to the rapid advancement in technology developments because you don't have access to the scanners or you don't have enough time to conduct those experiments in real life. I've demonstrated applications of visual imaging trials in quantitative CT. There are so many other applications. I think I showed a review article in the beginning of my talk, and there's also our website. So if you look at other applications, I encourage you to look at those publications and resources that helps you to show to see what's going on in other modalities and other applications. What is the future of VIT? Of course, there's still so much that can be modeled and improved. We can have more realistic and representative virtual trials to the extent and realistic enough so that it can predict patient outcome and can be used to advance an imaging question that we have. Of course, more applications are coming. We need to target diverse imaging modalities. I showed you results for CT scanners, but there's a lot of avenues, current work and needed work also in other technologies, and doesn't have to be also limited to imaging modalities. It could be other devices. I know there's a huge interest and great scholarship in the area of creating in silico devices also, and that's something that needs to also be continue to be developed. And then extending to treatment and outcome prediction, often we use medical imaging as a way to diagnose patients, but there's also huge interest and need to use it as a way to assess treatments or predict outcome. And if we can develop our trials in a way that would be able to predict such cases, then we can also use virtual trials for that application. So again, I would like to thank my colleagues at Duke University who collaborated and helped me with the material that I presented today. And if you were inspired by the work that I presented, if you like virtual trials, we are organizing a summit in 2024, April 22nd to 24th, in Durham, North Carolina. Visit our website. We would accept abstracts. There would be invited talks. There are student scholarships for traveling to the conference, and you would hear a lot about virtual trials in various applications and various developments from leaders of the field. So if you're interested, make sure that you check this out and make sure that you will attend our conference. And with that, I'm going to stop, and then maybe we can have some discussions and Q&A.
Hey, thanks, Ehsan. I really inspired with the work that you did in the past few years. I really enjoy. Now we are moving into a Q&A session to address any question or comment that the audience may have. As I mentioned earlier, please do type your question in the question bar. I can see some, but just giving the time to others to add their question. I'm going to start with my question. What is the level of interest in virtual imaging trial among the, let's say, the vendors, especially CT vendors, because you have mostly developed this new technology for the CT application?
Yeah, so we do collaborate with CT vendors, and we do work with them often. And I can say that there is a huge interest because they see various utility in it. From a design perspective, that's very helpful for vendors to have these trials, because before creating the hardware, they can test few things. There are various design questions that, let's say they want to do a new detector models, right? And if they want to study that and see how that would affect image quality, the old school way of doing it is that let's design some hardware with different materials and options, and then let's image them and see which one is better. But with this approach, they can basically virtually model that without a huge cost of manufacturing those, and study this. And do not study it in a simplistic way, but rather study it in for a patient population, looking at anthropomorphic conditions for various diseases. So that's one area that I think vendors are very interested, and there's a huge utility to it. Another area is that vendors can use a feedback approach of their current systems. So as I said, you can do protocol optimizations for various tasks. So vendors can look at these type of results and how it's working in a clinically relevant scenario, and then use that knowledge and insight to go back and improve their tools. That being a hardware tool that they have to improve, or that would be a image processing, image reconstruction tool that they can improve. So that's another area that I think vendors are very interested. And I would say the third one is regulatory. Often vendors, they design a new device and they want to show that it is effective, it is safe, and it has added value to the clinic. And it's really hard to demonstrate a tool like that, because also there's a lot of challenges of doing those type of trials for a scanner that is not still approved. In case of United States, like if FDA clearance or FDA approval are key, but then you cannot easily image any patients clinically for a device that is not FDA approved or cleared. So if you can use virtual trials in a way that would guide your and facilitate your FDA approval process, that is another avenue that vendors would be very interested and I think would benefit a lot from. Does that address your question? Any
yeah, excellent. Based on your answer, I can see that there's a great opportunity, and we will hear more about this virtual imaging trial in future. I'm just going to go to the question. First is from Hamid. Hi, Ehsan, and wish you the best luck of the webinar. Possibly it was at the beginning of the webinar. There is another one that, okay, sorry, it was a problem with the screen. Hopefully it has been resolved because we could see the screen very well. Another one is from Suranjana. Hopefully I pronounced that correctly. Hi, I'm new to this field. This question is with respect to slide 15. Can you point to the webinars or any textbooks, etc., that I can refer in order to understand how virtual models of disease are created?
Let me go back to, sorry, let me just quickly go back to slide. You said slide 15?
Yeah, 15. 1.5.
Okay. Okay, yes. Of course. So I mentioned some citations here. So for these diseases, you can see we have SPI for cardiac plaques. We have an AJR publication on COVID. We have a chest publication recently for emphysema models. There is this work on how to create lesions. Let me also show you this slide because I think that was also helpful. So if you go to our website, and if you can go to our resources and research, you can find various publications and various software and data that you can use for virtual trials. That's another good resource that you can use to learn more. And here there's this review article from Journal of Medical Imaging in 2020 that has a very deep summary and a review of virtual trials in general. It's not specific to CT. In fact, it has applications and discussions of other modalities and image processing aspects of virtual trials as well. So these are the, I think, good resources that you can look into to form your knowledge of virtual trials. So if there's any other specific questions, I am happy to answer that as well.
Thank you. There's a question from Yazdan. Thank you for great information. Is there a way to use Duxium? Is it open source?
Yeah, yes. So in our website, if you go, there is a Duxium is a resource that we would share with you. So if you can go there and then send a request, yes, you can use Duxium.
Thank you. Again, Yazdan. And is it possible to save the raw data by Duxium?
Yes. So Duxium actually saves the raw data for you. So then we also have reconstructions that you can use to reconstruct your data, but the main output of Duxium is sinogram images.
Next question is by Samueli. How many ground truth scan or ground truth scans do you need to create these cohorts?
I'm not sure if I understand the question. How many patients or how many scanners or how many scans?
Something that him or she mentioned is that ground truth. I think maybe it's referring to the data that you need for generating the population virtual patient that let's say cover the diversity that we observe in the reality?
Yeah, so on the patient side, of course, the more the better. So if you have more patients, it's always better. It highly depends on the application and what you're addressing. There is not a clear answer of me telling you 100 or 50 or 200. But what you want to make sure is that you understand the diversity that you're modeling. You know the spectrum of the feature that you're investigating, and to make sure that you have enough data that represent that feature. Let me tell you an example. Let's say you are modeling lung lesions, and the purpose of study is that you want to understand the effects of radiomics on in CT images and the effects of imaging parameters on accuracy of radiomics. So in that, you need to look at literature and understand the diversity of the lesion sizes. So if we have lesion sizes from 1 millimeter, 2 millimeter to 15 millimeter, and that's something that is documented, you want to make sure that you have that distribution. If we know that there are lesions that are round versus spiculated versus ground glass versus textured and all that, you want to make sure that you create those diversities. How many cases you need to create that, it depends on the diversity of the condition that you're modeling. But I think that the major aspect that you need to be mindful of is to understand the diversity of the problem that you're solving, and then make sure that you are representing it enough.
Makes sense. The next question is again by Samuel. You mentioned creating diverse cohorts. How limited are you when it comes to modeling disease, especially less common ones?
You are limited. It is more diseases is often more challenging because, as you said, you for the anatomy you get it from all patients. So any patients that you have, you can get the major organs. But disease, you need to look only a few cases that you may or may not have. So that's very valid point. It totally depends on the disease, but the good thing is that there are a lot of efforts in the research community that to generate such data, and this is very important and very impactful. Like for the COPD results that I showed you, there's this initiative called COPD Gene that they have acquired thousands of patients with COPD data, and then they made it available to the research community. Or like there is this disease called ILD, interstitial lung disease. It's a very rare disease. And then it's very valuable to have data to model it. So this is a very challenging task for us, but there is this community called OSIC, which they acquire data for ILD images of ILD patients. So it is a challenge, but there are efforts from the medical imaging community and other professionals to acquire such data, and then we would be always try to look into them and model them realistically.
Yeah, I think it's a big challenge because it's some virtual imaging trial is kind of the new technology, and no one has gathered the information or data based on the requirement of this. Thank you. Next question is from Saleh. Thank you for your presentation. Please let me know how consider or estimate variability level, uncertainty level of different random variables to make virtual patient.
Can you repeat the question, please?
Please let me know how you consider or estimate variability level or uncertainty level of different random variable to make virtual patients.
Okay, so let me see if I understand the question. So we do uncertainty quantifications in a sense that we want to understand, of course, when we are modeling things, they are approximations. And this is true for both the scanners and the patients. So we make approximations. So there is some uncertainty on that. So the segmentation that we make, or the assumption that I have for the physics of the X-ray scanner or the detector are not perfect. So the approach that we have done to make these authenticated quantification is that we do it in performing a sensitivity study. I didn't present it today, but basically what we do is that we target these parameters that we think might be susceptible to errors, and then how they would propagate and change any results. So let's say if we are uncertain about the lung volumes, we create patients at various lung volume shapes from the segmentation. Or let's say we are not sure about the X-ray spectrum. We create a library of X-ray spectra with slightly different estimations. And then we look at the final results, and that would give us some error bars. And with that, we can understand how our results are affected by those uncertainties. And if they are low, that's good. That's good news. We know that this is not a huge factors, and then that uncertainty would not result into an error measurement or error conclusion. And if they are, we go back and try to, in a feedback loop, improve our tools so that parameter would not be affected. I can give you one example. Like when you do CT simulations, you're modeling the source. Source is a continuous area. There's a focal spot area. In reality, you want to shoot your photons from that area. But computationally, that's not feasible. That would be a huge computation that if you want to model that. So what we do is that we sample the area, but then we are uncertain that whether that sampled version would create a reality or not. So we have done this sensitivity study and look at different ways of sampling it. And then going from like 1 subsample to 5 to 25 and so forth, and see that where that uncertainty would lead to minimum effect on the final results. And we've used that as a way to justify our approximations. I hope I addressed the question, but if there's other follow-up questions, happy to address.
Please. There's another question from Koning, hopefully I pronounced correctly. Is there any parameter fitting or adjustment involved when you're doing simulation? I think the phantom data with a virtual, oh sorry, my bad. Is there any parameter fitting or adjustment involved when you're simulating a phantom data with virtual CTs? How many free parameters exist in the computer models for the CT device, or are there no free parameters?
This is more of a physics approach of modeling it, so we are not really fitting any parameter. The inputs to to DuxSim is mainly the geometry of the acquisition, which are again come from the system that you're modeling. The common detectors you have, what's the distance from the source to the detector, the sizes, and all that. So there's the protocol. Then how do you want to image it? What's the dose level you want to use? What's the KV that you want to use? What's the bow tie filter that you want to acquire the data with? And then there is the interaction, which comes from well-known established physics models of how an X-ray would interact with a material. Again, Beer-Lambert law or Monte Carlo simulations with probability of an object and a photon being scattered, attenuated, go through, and all that. There is one parameter that we calibrate for, and that is a calibration of spectrum for one dose level, so we can calibrate the flux, meaning that I have an arbitrary flux and I need to know how much flux do I need for a particular dose, so I can get the close noise magnitude. But I do this for one dose level. But other than that, only one factor that you need to know for one scanner, which is a constant number. I cannot think of any other fitting parameters in our simulation. As I said, it's mainly based on the knowledge of the scanner acquisition and the physics of X-ray interaction with body.
Great. Next question is by Luca. Thank you for your great presentation. I wanted to ask, how did you choose your mathematical models to approximate anatomical structure or pathologies which are not well visible from current imaging technique? Can you demonstrate that your model are representing the reality if it's not can be well validated I think you touch it, but okay, we can add more comment.
Yeah, sure. So let me give you one example on the lungs, but this is applicable to other human models as well. So we want to create vasculature or airways. As you said, we cannot necessarily see all of it in the patient's image, so we need to have a mathematical model. The way we have done this is that there are morphometry measurements that address how these structures look like anatomically. So there have been studies that they have measured, let's say, airways or vasculature diameter, their branching angle, the wall thickness of them, and level of branching, and all that information. And they do it for a population of patients. For diseased ones, how does the branching look like for bronchitis versus asthma versus normals? And then in our mathematical model, we try to follow the same rules with some variability. So if there is in literature, like on average, the length of the parent to daughter ratio is about a third, then we try to make it the same way with some, again, variations to create this patient variability. And then how to validate it is that in the end, when we create these populations, we also look at those morphometrical measurements with our standard deviations and all that across various branches of the airways or vasculature, and we compare it against experimental studies. Or we can do this with high resolution macro CT images if they are available. So the basic answer is that we look at morphometry data or macro CT data, but then it's case by case depending on the structure that you're modeling and the data that you have.
Thank you. Next question is by Noef. Thank you for your informative presentation. Have you used any virtual imaging trial of metabolic bone disease using CT or MRI images?
Yeah, that's a great point. We have recently looked into osteoporosis and multiple myeloma as two major bone diseases. We haven't done it with MRI, we have done it with CT, and the idea is with the recent advancement of photon counting that you have spectral capability and you have improved the spatial resolution, how much of that would benefit more accurate quantification of diseases via CT. So we have done CT simulations of various scanners, particularly photon counting, looked at the quantifications of osteoporosis and also quantifications of multiple myeloma. So these are the two applications that we have looked at so far.
Great. Next question is by Sanjay or Sanjay. Sorry for my bad spelling. Do you contribute to IHC space IND compliance? Can you discuss in the respect of FDA? I'm not sure what those abbreviations stand on, if you know.
IND, you said?
Yes, it's a IHC space IND. You can check the, let's say, in the question bar.
I cannot see the question. Sorry, I was trying to see if I can see the questions, but it doesn't show it up for me. But I can address the FDA question that so far we haven't involved with the regulatory, but as I mentioned, there's a huge interest. There's a huge opportunity to be part of FDA approval framework using virtual trials. FDA itself has a great work with led by Aldo Badano and other colleagues there, that they are using virtual trials for regulatory purposes. We are also trying to go into that direction as well, to start using our tools for those applications. So there are actually avenues for it, and there are opportunities. So far, I have not been involved in that, but I'm very interested, and I know that other colleagues and leaders of the field have also done some work on regulatory aspects using virtual trials.
Excellent. And the last question from Janaki. Again, sorry for my bad spell. Thanks for the wonderful outlook. Could you share any first-hand experience on the using virtual imaging trial as a complement or substitute to conventional clinical trial for regulatory approval? Are the regulatory authorities welcoming?
Yeah, so as I mentioned, so again, this is not something that I have done, but I know that in the field of breast imaging, FDA itself is very involved and very active of using virtual trials and use it as a way to facilitate FDA approval process. In fact, FDA and University of Pennsylvania, they have demonstrated that for a trial, clinical trial, when they wanted to compare digital mammography versus digital tomosynthesis, another imaging modality of imaging for breasts, which they showed that they could have used virtual trial to predict that comparison that shows that digital mammography is better for calcification, and then digital tomosynthesis is better for lesion detection. There was a very costly clinical trial on that, but also FDA and University of Pennsylvania showed that you could have used a virtual trial approach to basically conclude the same answer. So there is a huge interest. There is a huge, I think, welcoming atmosphere from FDA. I think they appreciate the idea. They acknowledge all the benefits. But of course, when you approach for such applications, you need to make sure that your tools are realistic and validated, and they are predictive of clinical outcome.
Okay, thank you very much. With this, we're done with questions. Hopefully everyone has enjoyed. Ehsan, do you have anything further to say?
No, thank you everybody for joining. I hope you would have a wonderful weekend.
Thanks. Thank you again for delivering an exceptional webinar. So this concludes our today's webinar. Thank you everyone for joining us, and see you next time.
Goodbye, everyone.
Bye.